So we're going to talk about the topological insights on neuronal morphologies. And in order to do that, I will first describe what are the neuronal morphologies, how they're important for the rest of the networks and how they work within the brain. And then I will give some information about our work with Kathryn on the topic of neuronal morphologies. So here is a picture that we're kind of trying to put together all the pieces that we have information about in the brain. And we start from very, very small level, such as ion channels and the vesicles that we've seen topic earlier today, which are populating neuronal morphologies. And these neuronal morphologies create dynamical properties such as the electrophysiological traces that we can see. And together they form brain regions that create the dynamical properties of larger networks. And eventually they give rise to the network activity that we observe in the brain. But let's start a little bit from the broader picture and then we'll dive in. So first of all, what are the anatomical properties that are important for a brain? You see here two different pictures. On the left side, you see a picture, an actual picture from the microscope of a brain slice where you see the different brain regions. And what we're trying to do in the Blue Brain project is we're trying to reproduce as many details as possible computationally of these anatomical properties that we observe in the microscope and in the labs. So this is the left picture was taken in the lab where you see the different regions. And if you zoom in really into the details, you see the somata positions, so where the neurons are located in these different regions. And on the right side, you see a model developed by a PhD student in the Blue Brain, in which you recreate these positions within the brain. So once you have these positions, what you have is a density of neurons and other brain cells within the brain. But these cells are not really like points. If we delve into each one of these positions, they have these very distinct and complicated branching structures. And each one of these morphologies has a unique structure. However, what we can do to try to understand better this very, very complicated system is to categorize these morphologies into distinct categories. So we're trying to link properties that are shared among many morphologies to define some groups of morphological cell types that will give rise to their functional role within the brain. And starting for a very broad distinction, we have inhibitory and excitatory cells. So you've seen in the previous talk how inhibition and excitation can contribute to a large network. But here we're just trying to see what the different shapes of these two neuronal types are contributing into the whole picture. Once you have the categorization of the structural types, so the morphological cell types, then you need to go into the understanding of the dynamical properties of each one of these single neurons. And the dynamical properties is, if you simulate one of the neurons, you see different electrical responses. And here is just a small subset of example traces that you can get from all these different types of neurons. And just to clarify, we are in our lab in the Blue Brain project, we are only studying a very, very specific subset of the brain. So the work we have done is focused on the cortical neurons. And as you see on the right side on the C panel, these are the combination of different morphological and electrical combination of types that we can have into the cortex of the rodent brain. So you can imagine how broad this will be if we were trying to study all these types for the whole brain. So the question is, why are neuronal shapes so important? And to understand that, we need to understand that these shapes give rise to the electrical behavior of the neurons. So they define very specifically what the electrical responses will be from different properties of their shapes. A very simple example for that is you've seen all these different shapes in terms of branching. But what is not clear is that the thickness of these branches is having a huge contribution on how the neurons will behave electrically. So even the components that are not clear just by looking at the larger picture are playing a very significant role on how each of these neurons responds. In terms of connectivity, so in the larger picture, the neuronal morphologies constrain the connectivity because they define the connection points between axons and dendrites. So in order to have all these very complicated network dynamics that we see in the brain, you need to have these specific neuronal shapes that give rise to these different network dynamics. And this is pretty much the way that these two components, how neuronal morphologies shape the dynamical properties of brain networks on a large scale. And here is a very nice sketch, which is probably one of the first representation of neuronal morphologies by Ramon y Cajal. And that's how he started studying, looking at the morphologies and drawing them by hand. And now we are more at the stage where we can have digital versions of these morphologies. Here on the left, you see examples of different shapes of morphologies within the brain. And as I mentioned before, you can have very, very different branching patterns that we see. So this is somehow similar to the botanical trees that we see in nature. And as we have a very large diversity of trees in nature, we have a very large diversity of neural morphologies in the brain. So when I started my work in the Blue Brain, I was tasked with identifying different types of morphologies. And I was specifically given these two and many more layer 5 pyramidal cells. So those are excitatory cells. And the experts were telling me that they are clearly different. But when I was asked, like, how can you prove this mathematically that there is a difference? To my eyes, there was no clear difference. So it was very hard to understand what was different from my perspective, even though for the experts that were in the lab tracing these neurons, this was evidence. So what we did in collaboration with Kathryn and inspired with the topological data analysis methods was to find a way to encode the information that you see into this branching pattern into a topological description. And the motivation for that is that in the past, neuroanatomists were trying to measure specific properties. So they were measuring, for example, the length of the branches, the angles of the branches, the number of the branches. But all these are one-dimensional projections. So you were losing so much information from each one of these projections that you couldn't capture the full picture of the neuron. So what we were trying to do is to find a descriptor that would capture as many information as possible from these neurons. And the idea was, is coming from persistence homology. And the idea was to trace back each one of these branches and see how they connect to the root. And if we take as an example, so you can do that with many different types of distances, but if we can take as an example the radial distance. So here you see a very simple tree. And if you take any two of these tips of the tree and you follow it back towards the root, at each bifurcation point, you check between the two points. So, for example, 2 and 3, which one has the largest radial distance? And 2 is further away from the soma, so 2 persists, and 3 creates one bar here in the persistence barcode. So this way, we can follow back all the terminal points of the tree until we reach the root of which is the soma of the neuron. And this decomposes the neuron into these bars within the barcode, which represent the whole tree. An equivalent way to look at that is if we take the start and the end point of each one of these components within the tree, and we represent it in 2D. And here is like the start point is on one axis and the termination point is on the other axis. So each one of these points corresponds to one of the bars that we've seen before. And this is useful because then it allows us to average different trees, and by allowing Gaussian kernels around each one of the points within this persistence diagram. So this is pretty much setting up the background of what I'm going to use as tools to analyze neuronal morphologies. And here is an actual neuronal morphology, and you see this process of following back each one of these tips and decomposing this neuronal tree into the barcode. And as you see, we follow each one of the little smaller tips, and then we have the largest component, which generates the largest bar in the barcode. And as I mentioned before, we can take each one of these points and represent it into the persistence barcode. And then at the end, we can generate the persistence image by allowing Gaussian kernels around each point. So this is creating a summary that is capturing the branching properties of the neuronal tree. And we're going to use that for many different applications. Now, if you'd allow me to go a little bit into the mathematics, so we call it the topological neural morphology, so we'll refer to it as the TMD from now on. And we wanted to prove that the TMD is stable with respect to some perturbations. And these perturbations are motivated by the errors that we see in the experimental procedures. And one of them is the error in measuring the exact position of the nodes. So when you click and you make these reconstructions, you don't know that you got exactly the correct position. And the second one is you can have smaller branches that you don't see, or errors in the image that create branches that are not there. But those are the two types of perturbations that we wanted our algorithm to be stable against. And so we proved that the bottleneck distance between two pairs of trees that are with respect to these type of perturbations that I described before is bounded by epsilon, so by the size of these perturbations. So this was a very good example, a very good result, because then we can use this algorithm on the experimental data, even though they have intrinsically some of these errors in them. But the difference in the TMD that they're producing will not be significantly large, so we can use them without any big effect on the results that we get. So if we go back to the previous example and we perform all the actions that I described before, so we get the barcodes, and from these we represent it as persistence diagrams, and now we get the persistence images of the two trees. Now I think the difference is much more striking. So the experts were calling these type of trees tufted, because as you can see in the persistence diagram, in the persistence image, sorry, there is a tuft that is now very clear, and it represents that part of the tree. And the other type of neurons were the untufted, because this tuft is missing. So this is something we can now see very clearly with this decomposition into the topological descriptor that I described. But what is even more important is now we can actually do statistics on these type of descriptors. And one very, very simple operation is to illustrate what are the differences between these two trees. So in order to do that, we can subtract one persistence image from the other. And as you can see here, we get a higher density of branches at this location, which indicate the tuft that we've seen by eye. But interestingly, we see a difference also on the branches that are around the soma, which is not something that you can detect just by looking at the two pictures. So we have identified, for example, that here, the first effect is the existence or absence of a tuft, but also the branches around the soma have a different topology. So they're longer in this tree compared to this tree. And this is actually going to be interesting for a few examples that I will illustrate afterwards. So I will describe a few of the applications of these methods. So how can we use the TMD to classify cells, for example? To cluster them, and to finally for the inverse problem. And I'll start by the classification of rat pyramidal cells. So these are examples of excitatory cells within the rodent cortex, specifically rat. And the task was to find an algorithm to objectively separate these cell types, but also to try to explain what is different between these cell types. And here is an example of the layer 5 pyramidal cells that I described before. So here are just unique morphologies from these populations, but you see the persistent images of many, many cells, like 50 of the same cells. And as you can see, we have a very distinct pattern in which we have a tuft in some cases, in these two specifically. So these are large tufted pyramidal cells. Then we have the tuft that has a different shape, which is much smaller. And these are termed as small pyramidal cells. And then finally, we have pyramidal cells that are lacking completely this tuft, and they are untufted cells. So this way we could objectively classify the neural types within the rat. And we can also, this is a way to also understand what is different between the classes that we see. Here is an example. So in the cortex, we have all these cortical layers from layer 1, which is the superficial one, up to layer 6, which is the deepest one. And these are the classes that we have identified, and the names were given by experts. But as you can see, we get a very good classification accuracy for the main classes. Here is a more mathematically interesting example of we can use, so you see the different layers. So we're trying to classify the cell types, the excitatory cell types within each cortical layer. But what is interesting is depending on the distance that we use mathematically, we can have a very good classification, or we can have more vague results. And in general, the persistence images that you've seen before, so here, these type of persistence images were very, very good at distinguishing all the layers, and their differences could be identified, like we could pinpoint the differences between the different cell types based on the Gaussian images. As you can see, there are other type of distances, such as the landscape, based on the landscapes, that are also pretty good at distinguishing cell types. But the bottleneck, which is one-dimensional like projection, is losing information, so it's not able to distinguish cell types, so the accuracy is much lower in this case. Now, another interesting example is clustering. And the difference between classification and clustering is that in the classification task, we have the classes. So we want to see if we can objectively justify them and identify their differences. But in clustering, you have no classes. So it's like unsupervised learning. And the task here is we had some very nice neurons from human cortex. And we wanted to find if there is any distinct shapes within these neurons. And again, just by looking at them, it's very hard to justify that there would be 2 or 5 classes or whatever number of classes. And in fact, the experts here were not able to agree on which are the classes in which these neurons should be separated in. But if you do the persistence images again, you see a very, very striking difference. So you see that we have 2 populations that are very distinct in the human cells. Again, it's related with the shape and the existence or absence of these tufts. And as you can see, some of them are very slim. So we call these slim tufted cells, and the other ones have a very nice and profuse tuft, which we call profuse tufted. And what we wanted to identify is if it's a clear separation. So do we have these 2 classes, or do we have also intermediate classes in between? So I trained the classifier based on these examples, and then I extracted the probability that each one of these neurons that you've seen before, I think they're around 100, the probability that they fall within these 2 classes. And only 5% was not able to fall into these 2 classes. So that means there is a transition between these 2 classes, but it's very slim. So you see a very clear separation within this data set in these 2 classes. And what is interesting here, once we did this analysis, we were lucky enough to have the electrical traces of the same data set. So the electrical traces of each neuron that we've seen before. But once we did this separation based purely on their shapes, we saw that they exhibit unique electrical properties. So they fire at different rates, which is something we are still trying to figure out why this is happening and how these 2 types are different. But apparently the shape is important for their firing properties. So we use topology to group neurons so far. And now I want to show you a new example of morphological phenotypes of microglia. So apart from the neurons, we have other cell types in the brain. And one of them is the microglia, which are related to disease and lesions in the brain and the metabolic paths that you have. So here you see an example of different brain regions, and these are taken like photos taken from the microscope, in which the group of Sandra Sigurd that we have a collaboration with in IST, they took these images from the brain regions of the different brain regions, and they identified that the shapes of microglia in these regions are very, very different. And they could also reconstruct single microglia from these regions. But then the idea was to analyze them using persistence homology. So once again, we extracted the persistence images for each one of these regions. But here we have an important problem. So microglia are very dynamic because they respond to the metabolic pathways within the brain. They change a lot within very small timeframes. And the result of that is that we don't have a very specific shape. We have representative shapes and a high variation within these shapes. So the idea was to try to bootstrap in order the pool of microglia in order to create more robust signatures that would allow us to recognize differences between each brain region. And once we did this process, you can see here that the separation of the different shapes of the microglia for these different regions is very distinct. And this was something that was expected from this image that you see, because they look very, very different. So if you reconstruct each one of these microglia, you would expect to see a significant difference in their topology. However, this was not clear when you just study the shapes of single morphologies. As you can see here, they don't look that different. But what is even more interesting as a result is in this amazing data set that we were given from Sandra Sigurd and her group, there was a difference between, they were annotating the sex of the mice. So there was a difference between male and female. And if you do the same decomposition that we did for brain regions for one set of animals, so we put them together, but now we separate males and females, we see that the representation of each brain region in this hyperspace is very different. So what this tells us is that there is a different distinct pattern in the microglia within the brain, depending on the region, that can separate male from female microglia, which is interesting because I'm not showing that here, but they went deeper into this analysis and they studied how the microglia respond to different lesions. And what you could see there is that male and female microglia respond differently and recover differently for different conditions. So that could explain why some brain diseases are expressed differently in males and females that are related to the microglia shapes. And I think one of the main reasons to study microglia is because of Alzheimer's disease. They're highly related to Alzheimer's disease. So it would be very interesting to see what this connection would be with microglia shapes. And this brings me to the final application that I will present, which is the inverse problem. So what we did so far is to create from neuronal morphologies topological shapes and to use them in order to understand better the neurons from which they come. Now, what we want to do is, if we have these topological shapes, can we create neurons? Can we recreate the shapes from which they came from? And initially, that was a question of encoding. So if you have a specific barcode, can you get the exact neuronal tree from which it came from? But what we noticed is that this is not the case. So we cannot get a single tree that will, that gives rise to barcodes. So it's not unique to go from the trees to the barcodes and back. And Kathryn will talk more about this relation later on. But what I'm going to showcase here is we are using this inability to go back to single tree to create a population of different trees that are, each one of these trees is unique, but they have the same topological properties. So how can we do that? We start from the soma, and we start growing some like a random walk. So some type of directed random walk. And at each point, we have a probability to continue, to bifurcate, or to terminate. And if we couple these probabilities to bifurcate and to terminate to the barcodes, we can recreate shapes that have trees that have the same topology. So what we can do, we start from the root, and as the branch grow, a branch has a probability to bifurcate at each distance, which depends exponentially on the start points of these bars. So as you approach the distance from the soma for each one of these bars, your probability to bifurcate increases. And then it has very similarly a probability to terminate. So these branches, as you grow, are going to have a probability that to terminate that increases as you get to the end of these bars. So imagine if you only had one bar, it would start growing point by point, and it will just terminate at the end of the bar. So you will only have one neuronal branch that you could generate. But if you have multiple of them, this probability accumulates, so you can generate branching shapes that have very similar barcodes. And here is an example from the analysis that we did before, pyramidal cells of the rodent cortex. So here you see all the different types that we have of pyramidal cells that we have identified objectively. And in blue, you see the original cells. So these are random examples of the representative cell types. And in red, you see the synthesized, so the computationally generated. So use this method, and depending on the branching properties of each cell type, we could generate very, very different shapes. The important point here is that we didn't need to fine-tune the algorithm based on the different shapes that we see. So here, for example, you have these large trees with the very intense branching far away from the soma. But both of these and this one, which is super simple and it doesn't have any branch far away from the soma, both of them could be generated with exactly the same algorithm. The difference is captured in the topology that is it's sufficient to just have the barcodes of these different types in order to generate the different shapes that we observe in biology. And one of the reasons we want to generate these different shapes is because we want to create models of computational models of whole brain regions that are accurate up to the details of morphologies, at least. So what you see here on the right is an example of a cortical column in which we put together the artificially generated neurons, and the different colors are the layers the cortical layers that I described before. And as you can see, we get a soup of all these morphologies that is, and I'm going to show you later how we validate that, but it's very similar to the observed microcircuits that we have in biology. So we need to make sure that the neurons that we're computationally generating work correctly. So the first step, apart from the shapes that we just see by eye, is to electrically simulate them. And the idea here is that we have tools to go into the soma, induce some current, and see what are the electrical traces that they produce. And we perform these experiments for a number of different reconstructed and synthesized morphologies, and we see that they produce very, very similar traces. And this was just a single example, but we performed that into all the different morphological and electrical types that we have, and we also perform a statistical analysis for these in order to verify that all the different features of these electrical traces are correctly reproduced. Similar to that, we want to study the morphological properties of the cells. So we want to verify that if we generate from 50 cells of a specific type. So these are specifically layer 5 pyramidal cells. So if we generate from these 50 cells any number of artificially generated cells, if they will have the same properties. And here you see just a small subset of these properties in which the synthesized and the reconstructed cells have very similar behavior. So this is in order to make sure that the shapes that we generate artificially follow the properties of single neurons within the brain. However, that's not sufficient. So that's not really what we are aiming at doing. What we want to do is to be able to manipulate the barcodes in a way that we can generate shapes that we are not able to get examples from. And a very, very easy illustration of that is within the cortex, we have different cortical thickness. So we have different thickness as we move along the cortex. And this thickness of the cortex defines the size of the cells that live within this. So a very, very easy way to manipulate the cells is just to change their barcodes in a consistent way with respect to the cortical thickness. So as you can see here, we can start from a region in the cortex that's similar to where the cells were sampled from. And as we move along the cortex to similar sizes, we make the barcodes smaller and smaller, and we can generate cells of all the different types that have similar branching properties, but very different sizes. So we preserve the branching, but we change one component here, that is the size of the cells. So this might look very simplistic, but it's a starting point for something very, very for an interesting application that we're very excited about. So here is, again, a very, very simple modification that we're going to perform on the dendrites. So here is an example of an actual dendrite and the connectivity of the network that is formed in a column like this one that you see. So this is the connectivity that we get from a cortical column, and this is the connectivity between all the cell types that compose this column. Now, one of the modifications we can do is to take these structures and cut branches and generate a column based on similar cell types, but with smaller number of branches. And interestingly, what we see here is if we strip, so this is pretty simple to implement, because in the barcodes, we just keep the longest bars, and we start reducing the number of bars that we're using in order to generate synthesized cells. So interestingly, if we do that to all the cell types, and we end up with very simple dendrites, but the connectivity between the different cell types preserves pretty nicely, which means that even if you lose a lot of branches, the cells are able to connect to the different regions because the main branch is still reaching the cortical layers that it's supposed to. However, let's perform another very simple, again, operation, and that is shrink the dendrites. We take, again, all the cell types and we shrink them all at the same level. What you see here is even at, if I remember correctly, at 30% shrinkage, we were losing so many connections that we couldn't reproduce a network. So this is an indication that the way that you change the branching structure is going to have a huge effect on the network that these neurons form. And of course, as I said in the beginning, these are very, very simplistic operations on the on the dendrites, but our goal here is to try to reproduce specific dendritic lesions that we see in the brain, and they are associated to specific brain disorders such as stress disorders. So the shrinking of the branches was motivated because in many stress disorders, what they observe in biology is that the branches retract quite a lot close to the soma. So this could potentially explain how the networks collapse if the changes of the based on the changes of single dendrites. And here is a statistical analysis of the phenomenon that I described before. So you see that in case where the branches are cut, so you end up with having just the backbone of the neurons, the connectivity collapses much more slowly. In case where the dendrites are shrunk, what happens is that the connectivity collapses immediately. So you see a huge effect on the networks if you're shrinking the dendrites. And to summarize this part, so this is the idea to use topology to synthesize the rodent cortex. So we go from single trees to the topological barcodes and of each cell type, and we are recreating from this the cortical column that you've seen before. And even more impressively, now we can have the first brain region, which is completely generated artificially. And this is the rodent neocortex. So I'll pass on to Kathryn, who's going to talk more about the mathematics behind what you've seen so far.
Thank you, Lida. So yes, I'm going to take over now since I'm the mathematician, or the purely mathematician in this story. So I want to talk about how we can use these sorts of tools to learn something about how we can distinguish neuronal trees from other trees, because you could think about a neuronal tree as being some sort of combinatorial tree embedded in 3 space. And then you can say, well, is there something particular about the sorts of geometric trees like this that you get from when you're looking at apical dendrites of neurons? And if you want to think about, for example, different species, or you're thinking about different brain regions, are you going to see differences in the kinds of populations of neural trees? And can that be captured by this sort of topological signature that we're looking at when we consider barcodes? So you would consider, as is illustrated schematically here, your cortical column is being populated by barcodes rather than by neurons. And we want to see, is there something, so instead of studying the hard problem of saying what's so special about the tree structures that are shown up in neurons, you can make it more simple, more purely mathematical, and say, well, is there something special about the barcodes that arise from neuronal trees of this sort? So next slide, please, Lida. Right, so if you think about a barcode, so here we have a figure illustrating what we call the permutation equivalence classes of all barcodes with 4 bars here. So when you think about these permutation equivalence classes, what we mean is if you look at one of these barcodes, so we've arranged all of these barcodes so that the birth times of the bars, the beginning point of each bar, they're ordered increasingly. And then you can look at the other side to see what is the order of the endpoints of the bars. And this gives you a way of associating a permutation to each barcode. And it turns out that just looking simply at the permutation associated to a barcode, it actually contains a lot of information about your particular barcode. And it's interesting to classify the barcodes by their associated permutations. And we call these the permutation equivalence classes. And so one very simple representation of something as complex as a geometric tree, or the tree associated to a neuron, or dendrite in the case, is just the permutation equivalence class. So what permutation do you associate to its barcode? So next slide, please, Lida. And this gives us a way to think about how we can answer the question of whether this topological neuron synthesis that Lida was talking about, where you go from a barcode and in a stochastic manner, you're taking into account an exponential probability of bifurcating or terminating, whether this topological neuron synthesis gives you some way of having a kind of an inverse to the TMD. So what's illustrated in this figure here is on the top, the idea is you start with a barcode, and via this topological neuron synthesis, you're going to create some sort of a geometric tree. And then the question is, of course, there's a stochastic element here, so you're not always going to get the same tree. You're going to get, there's a sort of a distribution of possible trees that you can get. And then you can say, okay, I go from the barcode to the tree, and then I can again apply the topological morphology descriptor and get a new barcode. And I can say, how different is the new barcode I obtained from the one that I started with? On the other hand, you could start with a tree, a neuron, a dendrite, look at its associated barcode, and then apply the topological neuron synthesis and get a new tree. And you can say, okay, how different is the tree that I obtained in this manner from the one that I started with? And so what we did in work with Adélie Guérin, who is one of my PhD students, is to apply techniques from, well, not only topology, but also a bit of statistics and probability to be able to answer these questions. To what extent topological neural synthesis is some kind of inverse to the topological morphology descriptor? So the first question is, we formulate then in terms of what we call the stability of topological neuron synthesis with respect to bottleneck distance. So here, this is the idea of saying we start with a barcode, we look at an associated tree, and then we get a new barcode from that. So we start with the barcode, apply the topological neuron synthesis, it gives us a new neuron, and then we look at the associated barcode. And the question is, how different are the two barcodes, when you started with and then the one that you get from output? And the notion of distance that one uses between barcodes, so there are a number of possible distances. One of the most commonly used distances is something called the bottleneck distance, which is a sort of optimal transport distance. And so we said, okay, let's look at how far apart these are in terms of the bottleneck distance. Now, if you remember, perhaps from the description of the topological neuron synthesis, there's an important parameter in this construction, which is called lambda, which has to do with which parameterizes the probability of bifurcation or termination, depending on how far you are from one of the endpoints of the bars in the barcode for which you're constructing an associated tree. And the value of lambda that produces geometric trees that look the most like actual neurons is lambda is 1. So this is in units that have to do with the step size of the random walk that you're making as you create this neuron. And so if we focus here on the case when lambda is 1, then we're looking in this plot at the probability that the bottleneck distance between the two barcodes, the one you started with and the one you created by doing topological neuron synthesis and coming back down, is greater than epsilon. So when epsilon is very large, in the case when lambda is 1, which is the blue curve here, we see that the probability that the bottleneck distance is larger than epsilon is actually very small until you start getting to small distance with epsilon. So it's a sort of stability which says that the barcode that you're going to get if you go through this process, the new one, will in general not be too different from the one that you started with. So this is some way of expressing stability of this topological neuron synthesis. Next one, please. And this is a bit different, whereas another kind of stability that we were considering in the case where I said if you look at these barcodes where you have ordered the starting points so that they are increasing order like this, and then we had some sort of permutation coming from looking at the order of the endpoints with respect to the order of the starting points. Well, you could change that order by sort of switching the lengths of two of the bars. And what you want to see is also, if you're looking at this process of starting with a barcode, looking at the associated tree by the topological neuron synthesis, and then calculating the topological morphology descriptor, so getting a new barcode out of it, you could say, what is the probability that you're actually going to have two bars that change order like that? So again, focusing on the lambda equals 1 case, which is the most relevant case for us, we see that the probability that two bars will swap places like this when you go through this process of using topological neuron synthesis and then looking at the associated barcode, the probability that you will have two bars that have swapped positions, swapped orders in this process is actually, it's a function of the bars. So the greater the distance between the endpoints of the bars, the lower the probability that they will switch through this process, which is what you would expect. And so this is confirming this nicely. And particularly in the case that we're interested in, which is this biologically relevant parameter lambda is 1, then this the blue curve here. And again, we see that the probability of having a swap in order like that is quite small, as long as the distance between the bars is also very small, between the endpoints of the bars, more like. So as I said, it's sort of, you can go ahead, Lida. One of the motivating reasons we wanted to think about this was to say, well, what can we say? What is special about the barcodes that are associated to actual neuronal trees? So what we did was, for a very large number of reconstructed neurons, looked at both their basal dendrites, so the sort of root system of dendrites that's around the base of gather on the soma, and the apical dendrites, which are the ones that usually reach towards the surface of the brain, and looked at those as different neuronal trees and said, okay, well, let's look at their associated barcodes. And then we have something called the realization number of a barcode, which is how many combinatorial types of trees can have that particular barcode. So that's called the tree realization number of a barcode. And then we plotted based on how many branches or how many there were in the tree, or equivalently how many bars there are in the barcode, the realization number of these. And so in each of these, we're comparing, looking at the basal dendrites and apical dendrites. The red curve is saying what is the maximum logarithm of the realization number. So the largest, as a function of the number of branches or the number of bars, it's always n factorial. That's why we take the log here, because it goes very fast. But then to say, okay, but if we look at barcodes that are chosen at random, what sort of realization number are we going to get? And that's what the yellow-green points are here. So, and plotting that number of bars against their realization numbers. And then the blue points are coming from actual biological trees, coming from either the basal dendrites and apical dendrites. The basal dendrites seem to be more or less like random trees, but the apical dendrites are different. They have significantly lower realization numbers. So the number of possible combinatorial types of trees you could have with that particular barcode. So there is something special about the nature of these apical dendrites. And this came out in other ways when we were looking, for example, at the permutations that were associated to these barcodes and so on. So this really begs the question of, again, if we were looking at different species, if we're looking at different brain regions, if we're looking at what happens in disease, is this something that we could characterize or use in a way to distinguish these different states? So that was, I think, the last slide for me, indeed. So I think together with Lida, we would like very much to acknowledge all the wonderful collaborators we've had over the years. Lida and I started working on this together, these projects together back in 2015. At the time, we were working closely with Pavel Lutko, Ran Levy, and Martina Scolamiero on others. More recently, we've worked with my PhD student, Adelie Guérin. We would like to recognize Henri Markham, who's the head of the Blue Brain Project. And as I was looking at these slides earlier today, I realized we need to put some pictures up there of our collaborators at IST as well, because this most recent project with microglia is very exciting. It's really cool to see the difference between males and females coming out. But in any case, I'd like to take this opportunity to acknowledge the pleasure that I've had over the last 7 years in collaborating with Lida on this really exciting project. Thank you very much.
I hope you can hear the applause for a remarkable talk. Any questions from the in-person audience?
Thank you very much for this interesting talk. I have two questions. The first one is related to, well, both of them are related to rather to the first part, maybe to Lida. When you obtain those persistence diagrams from dendrite trees, did you ever, experience any problems due to the crossings, not bifurcations, but crossings which might appear between, for example, two branches bifurcating from a certain node? They cross each other and they continue still, and they terminate a point which is further from the point where they crossed each other. Maybe on two-dimensional let's say, figures or images of neurons. They cannot be distinguished, but we can imagine that in the three-dimensional space, in the real case, they might going over each other, and then they might still continue and terminate at a certain point. So I don't know if it was important for the morphological categorization which you made for the neural shapes, but
yes, so I can quickly reply to that because I did not go into the details of the process, but we are lucky enough to have three-dimensional shapes. We don't do it on the images, but we do the process on the reconstructions. So we have the three-dimensional shapes. So that's why we do not encounter these type of problems.
Okay, thank you. And the second question is, where you showed that this topological morphology descriptor idea is robust, stable against small perturbations. You showed quickly this theorem, where you mentioned that a certain kind of distance between two types, between the original one and the perturbed version. Well, a measurement of a global measurement of two versions of, let's say, graph, or graphs of neurons. What kind of distance it is, and can it be applied to no matter what kind of network or graph, or are there some certain conditions which make the use of such a distance function, such a global measure, available?
So the distance we used is the distance that Kathryn mentioned. It's the bottleneck distance, and it's a topological distance. To define the distance in the space of trees was kind of hard. That was the main reason we tried to do it in the topological space. So this is related to the persistence diagrams, this distance, not to the trees directly.
And there are many known ways to define distances between trees and between embedded trees as well. But because the idea was to look in the end at these topological signatures, which are the topological morphology descriptors, it was relevant to use some well-known and well-studied notion of distance between the barcodes, and then to use some relevant, biologically relevant notion of perturbation, of having two geometric trees that are relatively similar to each other. And that's why that was the way we formulated this result. But there are other notions of distance one could use that can be more relevant in other contexts.
Yeah, thank you for the lovely talk. I want to ask, I think you did talk about this, but I wasn't completely clear about how your methods take it into account, which is that it seems like the most important aspect of neuronal morphology, and this is certainly true in the retina, is which layers are connected, from one to the other. And so I wondered how you're taking the layer into account in your initial barcode, and whether you showed very beautifully that that by scaling, right, by the thickness of the cortex, for example, you can get dramatically different changes. So do you always take into account layer structure in the original setup of the coordinate system?
Yes, that's a very important point indeed, that we have information on the cortical depth for each neuron. So we know up to some uncertainty the layer from which the neurons are coming from. So this is indeed a very important parameter in the way that we construct the column and in the way that we analyze the data coming from the lab.
I guess it would be cool, right, if your heat maps of shape sort of pulled out which layer is connected to which layer, where it originates, where it tufts. So a distance along that diagonal, for example, if that told you which layer, because for the retina, what you see with the retina is that it just tufts, straight right into two different layers. And so If you clustered retinal neurons that way, retinal ganglion cells, they would just be sort of points in a two-dimensional space according to which layer was connected to which. But in your measure, you don't take that into account directly in the barcode. Is that right? That you can't see any layer structure directly from the barcode.
Yes, so we don't take that into account. If you do it for the excitatory cells, you can separate layers because primarily because of their sizes. But for inhibitory cells, you can't really tell the layer just from the barcodes. So this is a component that we add externally. It's not within the barcode.
Any more questions from the audience?
Yeah, sorry. So I have still a question about this choice of the distance, so the bottleneck distance. So I understood that since it is a well-known distance and well-studied distance, it's natural to use it. But so I'm not really familiar with the data, but I was just wondering, since the raw data you have are trees, why not use directly a tree distance rather than bottleneck distance, which is more associated to the topology? But because from my point of view, these trees in our topology, they are used to describe a more complex structure. So it's just a way to reduce the dimension of the problem. But for you, the data are already trees, so why not use a tree distance? Maybe it's a stupid question. I'm
Are
sorry.
you referring to the stability theorem that we were discussing, or in general? Because in general, so there are very different answers to that. In the stability theorem, we wanted to see if you have different trees, how different the barcodes that will be generated. So that was the reason we used a bottleneck distance. In general, in the classification tasks, we are not really using bottleneck because this is a very simple distance. So it does not give enough information, as you correctly pointed out. But we're using distance between the persistence images. We are not using distances directly on the trees because, first of all, they're computationally very, very expensive. So the most well-known distance is the edit distance between trees. And if you try to compute that, it's supposed to be NP hard. So there is no algorithm to do that. And in addition, the edit distance is not taking into account the geometry. It's just taking into account the decomposition of one tree into the other by changing the branching structure. So this was not something that was easy to use directly. And this was actually the reason we made this type of decomposition into the barcodes that are much easier to study, and there are well-known tools to study distances between them.
All right, thanks.
There is at least one more question from the audience.
Hello, thank you for this very nice talk. Maybe I missed it, but I was wondering if the synapses were important in your study. I think because it's important for the connection between the layers. And if you take it into account really in the analysis, and how you would place the synapses on the tree.
So in our analysis, we do not have this information, so we don't get, unfortunately, the locations of the synapses. This is something we have been discussing with a few different experimental groups to try to see, for example, spine locations, and try to see what are the differences for spine locations, but for synapses, we don't have this information. Now, for the resulting circuits that we generate, this is a completely different team in my lab that is doing that. So this is a different team in the Blue Brain project that they're assigning synapses and then all the synaptic properties. So I'm not at all an expert to talk about that. We do have it in the network models that we generate, but that's not something that I worked on.
There is one more question from the audience.
Thank you. I'd like to know how the molecular classification overlaps with the morphological classification, because in the cortex you have multiple inhibitory subtypes like somatostatin, VIP neurons. Can you tell what is the molecular subtype just looking for the morphology?
So that's a very good question. And that's the direction we are going to at the moment. So we're trying to link transcriptomic information. And there is a very good data set from the Alan Brain project in Seattle, in which they have transcriptomics with morphologies and electrophysiology. So we are going to use that to see how the phenotype correlates with the transcriptomic and molecular information. We have indication that for some big classes that's the case, and we can see, for example, Martinotti cells being very well separated from basket cells, which have different molecular signatures. But I can't reply yet for every molecular information that we can get from this analysis, because we haven't finished these analyses yet.
One last chance to ask questions. If not, let's thank Dr. Kanari and Professor Hess for a great talk.